Cancer Research Center. Our moderator for this evening's event is Dr. Eric Holland, Senior VP and Director of the Human Biology Division at Fred Hutchinson Cancer Research Center. Please offer up a very warm welcome for our panel tonight.
So, we're happy to be here, and we've discussed amongst ourselves, I'm Eric Holland, by the way. We've discussed amongst ourselves what we thought would be a good format for this meeting. And we thought it would be something like each one of us had a topic in this space that we would discuss for 5 minutes or so, just to get things going. And then to open it up for questions and ideas. And we may need to prompt some of the early questions on our end, but we'd love to just start it going here. So each of us has picked a particular topic in this space. And the one that I'm going to talk about, and I'll talk first, is the issue of personalized medicine in cancer and how this relates to systems biology. So this is something that is a hot topic these days, and it's a phrase that means a lot of different things to different people. So one of the simple things that it means, and I guess I'm going to describe 3 different ideas of what personalized medicine could be in the cancer space. So the simple version of that is that each of these tumors that we might have frequently will have mutations in a particular gene. And you could say, well, this is a lung cancer, and so it might have mutation in gene X, whatever that would be. But the truth is that although there certainly are some lung cancers with a mutation in gene X, there's mutations in a lot of other genes in a given tumor, and across, if you look at a lot of different tumors, you have this tumor might have a driving mutation in gene X, and that might have a driving mutation in gene Y. And so trying to find the mutations in your tumor, and if those mutations are actually druggable, in other words, there are available drugs currently that would target that particular gene, then that is one version of personalized medicine. People are going to take your tumor, try to find the mutations that have a drug associated with it. And this is not just fiction. If you take lung cancer as an example, there are maybe 25% of these tumors have a mutation in a gene called EGFR, and there are drugs against EGFR, abnormal growth factor receptor. And a much smaller percentage have a mutation in a gene called ALK, and there are ALK inhibitors out there. And in fact, the trials show improved survival in patients that are selected to have those mutations and given those drugs than if they aren't. And so, that's one version. Okay. So the second version of personalized medicine is, if you look at, I was thinking of showing slides, but I'm just doing it with my hands here. If you imagine what we refer to as a survival curve. So you start here with 100% of the patients still alive at the beginning, and as time goes on, fewer and fewer patients are still alive. And obviously this would be a good curve because everyone stays alive. This would be a bad curve because everyone dies right away. And the reality is that the curves frequently look like this, but there's a tail at the end in many of these trials. 5 or 10% of the patients actually seem to do well on drug X. And if it's only 5 or 10% of the patients, then that drug X doesn't actually that's a failed trial, because that's not enough patients to allow the drug to be labeled by the FDA. But that doesn't mean that 5 or 10% of the patients didn't actually respond well to that drug. In fact, it would be great if you knew that you were one of those 5 or 10% that drug was on the shelf in pharma, because it didn't pass the test, but it was your drug, right? So, the second version of personalized medicine is to try to take these rare responders, these people who actually seem to respond well to drug X, and try to figure out why they are that way, to molecularly characterize them and just define them in a very granular sort of way, such that if you turn out to get that tumor, then that drug that they got is a good drug for you, right? And so this is not necessarily looking for one mutation. It may be looking for a collection of different things which describe that kind of a tumor. And so that's the sort of the second version of personalized medicine. And the third is maybe a little bit even more complicated. So imagine this, that if you have, imagine we all have a tumor of the same name, right? But every single one of these tumors is a little bit different, just like everybody's nerve is different, which is true. And just imagine that where you sit in this room and who you're next to is a reflection of how similar your tumor is to the next person. So your tumor is actually quite a bit different than this person's tumor, but very similar to the next one over. And if we could do this for a whole reference group, and this was in a database where each one of your tumors and molecular characterization of that tumor, and your entire clinical story as to how you responded to drug X and how long you lived, and all these various things about your clinical course were part of that database, then the next person who comes through that door right here and has their tumor analyzed, and now we can say, well, you actually belong over in that neighborhood. And now we can say, well, how did you guys do over there? Did you respond to drug X? Did you respond well? Did you live a long time? Or even, the ones that lived long, what did we do to make you guys live long? And the ones who didn't live long, let's try not to do that for these guys, because that actually might be different than this group over here. And so instead of saying, you all have glioblastoma, or whatever the word you're going to use, you actually have this neighborhood, because that's actually what's most similar. And so that is an ongoing effort as well. But that requires a lot of, that's big data, right? It requires a lot of people, a lot of numbers. For each person, it requires a lot of data for that particular person. But this is the kind of big data in systems biology that we're in the time now where this is happening. In fact, we have the ability to just met with someone today who has a database of 220 million people, almost all their buying habits and everything they've done. Just this huge database that it's really designed for marketing. But the truth is, this could be used. Who's to say that maybe you're buying something off the shelf, some sort of nutraceutical that's actually affecting whether you're responding well to a drug or not. And so you can imagine going to the next level and really bringing it all together and really in sort of an Amazon sort of way, figuring out, what's going on with you. And so that's the third version of personalized medicine, the one that really brings systems biology sort of full force. So that's the one that we're particularly interested in the tumor that I have my background in, which is brain tumors, because we don't really have any targeted drugs that work against a mutation. And there are some rare responders, but they're really rare. And so this other sort of use of big data is something that I'm particularly interested in the type of tumor that I worry about. So that is my version, my 5-minute story, which might have been more than 5 minutes. But I'm more than happy to entertain or begin the discussion, which we'd love to have people just give ideas, talk about things, and if you have ideas, come up to the mics, and because they want to record your questions. I could restate them, of course, but we have a question over here. Yeah, if you stand up or walk over to the mic, maybe. And I know that's kind of a pain, especially if you're buried in the middle of the neighborhood over here.
Hi. Can you talk a little bit about the interface between personalized medicine and the sort of problem that people talk about in terms of the orphan drug industry, where we have all these people here, and you've done your tests, and this gentleman is the only one who responds to the drug.
That's right.
To treat him is going to cost $500 million a year. Who's going to pay for it? And does that affect at all your approach to medicine in terms of how much does it really cost to make everything personalized, which almost sounds like unique and individualized, and obviously very expensive for someone to have to pay for it?
Right, that's a great question. So you were referring to drugs that are on the shelf developed by pharma, and he's absolutely right that the number of dollars required to develop a drug is it goes up every year, but it's in the $1 billion range, something like this. It's way too much. Which is, why some of these drugs, cost tens of thousands of dollars per month after you actually are given them. And you can certainly imagine that this is a non-sustainable concept. There's no question. You know, I don't really know. I mean, we can have a long discussion about why it's so expensive to develop drugs in the United States. You know, this is a great topic. You know, I'm not sure that's what the topic of this meeting is about. But it's expensive. There are a lot of drugs on the shelf, and because it's so expensive, pharma tends to, you know, only really invest and go full force in something that they really think they're going to make their money back on. And so when you have, you know, drugs out there which may very well be good for your tumor, and you're one of the very few people that have it, they aren't going to invest $1 billion for your drug if it's just you, right? They're just not going to do it. So, you know, it would be wonderful, and that's what some of these rare responder types of trials are, and they are basically to identify drugs that may be sitting on the shelf of pharma that could be pulled off the shelf under certain circumstances, and we could try to use them if we sort of knew who that drug would respond to. And there's a very famous drug called Gleevec, which is one of the very first personalized or targeted therapies that was sitting on the shelf in Novartis, and they weren't gonna go forward with it until some scientists you know, begged them to pull it off the shelf, and they realized there was a particular relatively rare tumor that this drug was actually good for. And once they realized it was good for that, then of course everyone wanted to use it, and you know, they actually made money on it. But yeah, no, I think this is a cultural problem we have. It's a financial one, but I should also say that you know, some of these personalized things aren't necessarily have to be drugs that are you know, billion-dollar drugs. I mean, there's a lot of drugs out there that are already past the patent time, 20 years, and they're relatively inexpensive, and they're choices that the clinicians make. I mean, do you give this drug versus that drug? And they're not necessarily that expensive. But it could be that these off-label drugs, or not off-label, these drugs that are not these really expensive high-end drugs, but older drugs, actually could be chosen as a better choice for you if you were somebody who would respond better to them. And so personalized doesn't necessarily have to mean expensive. I guess that's the long answer to your short question. Yes?
Could you just define what systems biology is?
Well, the inventor of systems biology is sitting right here, so maybe maybe I should. Yes, he's going to define it. It's big. It's big. It's a lot of things interacting with each other. But I think that's his whole thing. So I'm going to defer to his topic. That's it. Wow. Go on to the next one. So the next one is Dr. Pete Nelson, who is a urologist and a heck of a nice guy from the Hutch.
I'm actually a medical oncologist, but close enough. So I'll give you a little bit of perspective. From my standpoint as a medical oncologist, the three cancers that I primarily see and treat really gives us a remarkable spectrum of cancer behavior and why we need to understand and individualize treatment approaches in a much more thoughtful and thorough way. So the three cancers that I primarily see, one of which we study in the laboratory, is bladder cancer, testes cancer, and prostate cancer. So bladder cancer is quite interesting because if it's found very early, you can rev up the immune system through very non-specific ways and completely eliminate those superficial cancers. But once bladder cancer spreads and gets into the bloodstream and goes to distant places, it's essentially incurable. In contrast to that, testes cancer, which you're probably very familiar with Lance Armstrong's story, even when it spreads widely throughout the body, even into the brain, is very curable with types of therapies that we've had for 20 years. Very non-specific, very non-targeted. And then the third malignancy, prostate cancer, is almost ubiquitous. If a man lives long enough, basically every man will develop prostate cancer. That tells you the vast majority of these we never want to detect. We need to understand the variation inherent in those prostate cancers and determine why most will remain indolent, whereas others will spread and be quite lethal. But even when prostate cancer spreads, it's dependent on what I call a molecular engine, the androgen receptor. So that's probably the first example of a precision medicine target, and that is blocking that receptor or eliminating its fuel, which is testosterone, puts the vast majority of these men into complete remission for a period of 2 to 3 years. It's not a cure, but it's a remarkable response that you won't see in any other type of cancer. So that tells you that we have 3 cancers here that I simply see in my practice that behave very, very differently, which tells us also that the molecular foundations or the underpinnings of these cancers are also quite different. So there's been a big effort through the National Cancer Institute over the past 5 years to build on the Human Genome Project. So Lee Hood, you'll probably hear from a little bit later, his role in sequencing the human genome, which provided a foundation really for the big data that we're talking about. That's really a scaffold of both normalcy as well as the inherent variation that allows each of us to develop and respond to our environment in different ways, and it also dictates susceptibility to cancer. So that scaffold has now been built upon by sequencing and determining the molecular changes that happen in the common cancers that affect man. So there's been this big effort called the Cancer Genome Anatomy Project to define the mutations, the genomic alterations, and they've primarily been focusing on, again, the common human cancers and sequencing about 500 just to try to get the landscape or the full spectrum of the changes that occur. Not initially focused on drug development and therapeutics, but trying to determine what vulnerabilities might there be. So our role most recently has been in the laboratory to look not just at the localized prostate cancer, but to define the molecular or genomic or big data landscape of prostate cancer once it's spread. This is a difficult task because it takes biopsies of metastases. What we hope in the future is to be able to determine this molecular landscape from non-invasive or less invasive ways, for example, tumor DNA that may be in the blood or that you can pick up in the urine of tumors that might be shed. So that's really the hope in the future. With this large prostate cancer genome project, if you will, we do have several clinical trials built into it to test this idea of precision medicine. So in addition to that androgen receptor, which I mentioned was probably the first precision medicine target, we've identified several other molecular changes that point to specific vulnerabilities for drugs. So Dr. Holland mentioned that occasionally we find alterations in one tumor type that are also found in another tumor where there's already a drug developed. So an example of this that we found in prostate cancer, the breast cancer susceptibility genes BRCA1 and BRCA2 point to a particular vulnerability to a type of a drug called a PARP inhibitor. And we found now that about 5% of the men with prostate cancer also have this particular molecular change. So there is now a clinical trial designed to test whether those PARP inhibitor drugs will be able to exploit that specific vulnerability. So that's an example of how we're looking through the entire genome of these tumors to find vulnerabilities. There's lots of issues as well with hereditary prostate cancer where it will be important to bring in the big data of understanding predisposition that will be hardwired in the genome but then modified by environmental factors. So I'll end there, and we can probably just take questions maybe after all three, because I think they want to hear what systems biology is all about.
So my name's Tony Blau. I'm at the University of Washington, and You know, 5 years ago, I wasn't in the cancer field at all. I'm a hematologist, so I came to Seattle because of bone marrow transplantation 25 years ago. And 25 years ago, doing our best for a patient with cancer meant really giving them enormous doses of chemotherapy and radiation, getting as close to the brink of death as you could, trying to kill the tumor cells just without killing the patient, and then transplanting them with someone else's bone marrow, and strapping in and writing out a really rough time. And while that clearly was kind of the example of what it meant to do our best 25 years ago, I believe that today, perhaps no patient with cancer gets what we could defensively call our collective best. And I think the reason for that, you could think of as on a single graph, where if we were to plot the rate at which technology is improving, it's breathtaking. It's explosive, it's exponential. And if you plot the rate at which those technological advances are being applied to the treatment of cancer patients, it's improving, but that's linear. And so if you were to put yourself in the position of a patient with advanced cancer, you're facing this ever-widening gap between what's possible technologically and what's actually brought to bear in your care. And that's, in part, so I think the limitation is not that we need a new technology or we need a new series of technologies to fundamentally advance cancer research and treatment. What we need, I think, is a better way of applying the technology that we already have to patients with cancer. And so toward this end, we developed this thing called the Center for Cancer Innovation at the University of Washington. It's based at the University of Washington, but it has over 100 investigators from about a dozen organizations. And it's really built on two big ideas. The first is to find researchers who are experts in their domains, from many different domains. Cancer is an incredibly complex problem, therefore you have to find ways of harnessing the expertise of people from very diverse areas of science, technology, and medicine. And you have to focus your efforts not on cancer in the abstract, but cancer as it exists in a human being. So the other thing you need are patients with advanced cancer who want to be the subjects of an unprecedented scientific investigation. And in our clinical trials, we've initiated our first clinical trial in a type of breast cancer called metastatic triple-negative breast cancer, so a form of breast cancer that's generally incurable with conventional therapy, where these women allow us to do multiple biopsies of their tumor. Not only is every cancer different from any other cancer, but within a patient, the cancer is continuously evolving. And so if you really want to capture the status of a cancer at a single point in time, you can't just get at one piece of tissue. You have to really sample it from multiple different sites of the body. Collect this tissue, sequence it, digitizing the patient and her tumor. So collecting billions of data points, placing it on the cloud, and making it accessible to some of the best computational biologists in the world, who bring their novel solutions to try to how to integrate that vast data set into an accurate understanding of what's going on. And the problem is, when you have enormous amounts of data and only one patient, there will be thousands, if not tens of thousands, of seemingly plausible ways of putting the data together, the vast majority of which are wrong. And so how do you find that accurate solution out of this vast background of noise? And so the idea is to find the best people that you can find in the world and have them do their best, to make their best, give it their best shot to try to figure out what's going on in that patient. Look for a point of vulnerability that can be attacked with a targeted drug, and then test your prediction in the patient. Find a way to get the patient on the drug that you predict to be effective, and then use that patient's outcome to feed back the information back to the scientists and the computational people, creating a learning loop. And rather than trying to search for a one-size-fits-all treatment for cancer, which we don't believe exists, this model is one of all for one, bringing the world's expertise to bear on the problems of an individual cancer patient, and one for all, using that individual patient's experience to teach the world about how cancer works. And what we're really, and it's so important in this model that we be incredibly transparent about how primitive our ability is to convert these vast data sets into accurate understandings of what's going on in the patient. We're just at our infancy in this process, and so our predictions are certain to be wrong many times. And it's so important that we be transparent about that. And that the patients that are in this trial are making a heroic contribution to advancing the boundaries of cancer research. But what we're really aiming to do through this process of what we call intensive longitudinal monitoring with multiple biopsies at multiple time points over the course of the patient's illness is to create a vast database, which we believe, as we enroll tens, hundreds, and eventually thousands of patients, will amount to an instruction manual for how cancer works. Analogous to the time that I was an intern now many years ago when HIV was rampant. The first HIV drugs, AZT, came along, and when we gave it to our patients, the virus would disappear, but then it would come back having mutated. We'd give it the second generation of drugs, the virus would disappear, and then it would come back. And over the years, we've learned the rules that HIV uses to evolve and escape treatment. We know what the rules are for HIV, so that if you get HIV now, you get drugs that not only stop the virus, but that also block off the anticipated mechanisms of escape. And so we need this rule book for cancer. And I think this is going to be a very, initially, certainly a very expensive process. I mean, is it really, it's a paradigm shift to say the least, that you're going to take a lot of money and spend it to go deeply on few patients rather than spreading it out over many patients to have a more shallow look at what's going on. It's by no means consensus about that's the right way that the money should be spent, but that's what we believe. And then I think it's going to take a long time, and it's going to be heartbreaking. But I think that we're just at the inception of what is a multi-decade effort that I think ultimately will allow us to really make dramatic progress in cancer. And with that, I'll turn it over to Lee Hood.
Well, I guess I have to define systems biology. So, when I was a young assistant professor at Caltech in the early 1970s, what I really became impressed with was the enormous complexity of both biology and medicine and disease. And that complexity is absolutely logical because life is driven by this process called Darwinian evolution, which is a random and chaotic process. And as a consequence, it doesn't build biological entities using the law of Occam's razor in the most efficient way. It builds them as contraptions where one success is built on past successes, is built on still previous successes. So it's very much like, I mean, imagine a Rube Goldberg gadget where you tied together 14 different things that allowed you in a clever way to cool your soup. So if you wanted to analyze that really complicated gadget, what would you do? And this is the essence of what systems biology does. One, you'd have to define all the parts. Two, you'd want to find out what each part did individually. Three, you'd want to understand how those parts were connected together. Four, you'd want to understand the dynamics. How did they move to eventually cool the soup? And with that, you'd want to understand the function. That's exactly what systems biology is. So fast forward to cancer and other diseases. I would say medicine has failed in really fundamental and interesting ways in studying disease. And there are two aspects of disease we don't study virtually at all. So one aspect is we don't study the earliest origins of disease. We don't see what happens when you diverge from that path of wellness to a path of disease. And of course, the second thing we can't do in human patients easily is to study the progression of the disease from its initiation all the way to the end. So we've thought a lot about how could we change this fundamental limitation in medicine. And of course, this generalization holds for cancer, but it holds for all other diseases as well. And the conclusion that we came to is the only way you can achieve these ends is to create a longitudinal Framingham-like study. This is a famous study that started in 1948 with 5,000 patients on cardiovascular disease, and it's continued up till today with lots of measurements every year. But we want to do it on 100,000 well patients, and we'd like to do it again for a 25 or 30-year period. So what happens if we think about doing such a study on 100,000 patients? You'll see patients gradually segregate into one of two groups. One group will remain well or improve in health. The other group will begin to diverge into disease. And of course, what we're interested in is the group that will diverge into disease, because what we're going to do with these 100,000 patients is make a whole series of longitudinal measurements all the way across the duration of time in which we study them. So what that means is at that earliest point of divergence, say to cancer, we're going to have a lot of data that we can look in retrospect back on, and we'll be able to do two things with that data. One, in time, we'll be able to develop extremely effective diagnostics that pick up those very earliest changes. And number two, we'll be able to study the mechanism of that early initiation of disease. And the hope is that you can then design drugs that will take that patient from the disease path and put them back on the wellness path and prevent that whole vector of sickness and save the healthcare system an enormous amount of money. So the assays that we're talking about, we'll do a genome sequence, we'll do your gut microbiome, we'll do clinical chemistries, we'll do quantized self-measurements of sleep and heart rate and respiration, all of those things. We'll also do fingerprints that exist in the blood that are specific for the brain, the heart, and the liver that will give us very early indications of transitions from wellness into disease. And we'll do many of these measurements 4 times a year, so you'll have an enormous amount of data. Of course, the real challenge, and that's one of the grand challenges of systems biology, systems medicine in this case, is how do we take that data for the individual and aggregate it together and then mine it and finally integrate it to create models for the patient that will delineate the actionable opportunities of how you can improve their health. And that is the biggest challenge in these studies, apart from getting the money to do the studies. So what exactly do I mean by actionable? Well, in the case of cancer, it turns out that there are some cancers that are genetically determined. There is a syndrome called Lynch's syndrome that is adenocarcinoma of the colon that occurs in about 3% of the patients. If you have a single bad copy of that gene, you have an 80% chance of getting colon cancer by the time you're 50 years of age. So if you know you have that gene, is it actionable? And the answer is yes, because basically what you have to do starting about 30 years of age is every year have a colonoscopy, and if there are any little early adenocarcinomas coming up, they can be clipped off, and people can be kept healthy in exactly this way. So that's one aspect of the actionable. The other part that will help us begin to understand cancer, of course, is as we accumulate this data, then we can begin to understand retrospectively for given patients with cancer just when it was likely they diverged, and we can analyze those data, again, for the early diagnostics, for understanding mechanisms, and hopefully how to convert things back into a more normal state. So, how are we going to proceed with this? Well, we're starting actually this month with the first 108 volunteers that are beginning through this study. And we're going to carry it out about a year. We're going to learn everything we can about how to put this study forward, because in the next year we want to move to 1,000 patients, and then 10,000, and finally to 100,000 different patients. So I would argue that this study is the opening gambit of an enormous revolution in healthcare that's going to move us from a reactive medicine that's focused on populations of patients, that is interested entirely in disease and not on wellness, to one that has a real focus on wellness, that has a focus on understanding disease transitions, and really transforming healthcare by making it possible to reverse trajectories very early on in the disease course. So I think with that, I'll close a new idea on how we can deal with cancer. And I think what we should do is open it up to general questions for all four of the speakers. And again, it would be really terrific if you'd come up and speak in the microphone, because everyone can hear you then and they can get the recording. So please.
How do I sign up?
Well, we are beginning to take lists for the next tranche, the 1,000. So we have people from ISB here that you can talk to, and they can help you get signed up. Are there questions on cancer?
Yes, could you speak to the role of epigenetics in cancer and how that interrelates with the genetics?
So epigenetics, so in the old days, we used to think that the genome was immutable. You got your genome early in life, and that was it. It never ever changed. And it turns out that it can be changed in two interesting ways. And these changes are called collectively epigenetics. And the most typical kinds of changes that occur in epigenetics is you turn off the ability to express certain kinds of information, so that changes what you can do. But the changes occur on the genome, so the genome is dynamic, just like the proteins in your body and the metabolites in your body and all the other things are dynamical. So everything is dynamical, so we have to come to understand how the genome changes, as well as all of these other things. And it's clear that the epigenetic changes play a major role in development, and they play a major role in a variety of diseases. Yes?
Is this study of 100,000 people just for cancer, or is it for all diseases?
It's for all diseases. I mean, the idea of starting with 100,000 people is that you'll see disease transitions for all the major classes of diseases. So you'll see cancer, you'll see cardiovascular, you'll see neurodegeneration, autoimmune, and all the rest. And you'll be able to look from a single study at all of these divergences. How about some speakers for other people here?
Well, I'm sorry, but I do have something I'd like to address to you, Dr. Your emphasis on systems biology is absolutely terrific. I wonder if in your thinking you've included the role of the behavioral and psychological sciences such that people's thoughts and feelings and behavior enter into health and disease, and especially when you want to incorporate positive health outcomes. A lot of times it's the person's thoughts and behaviors that puts them, like people here that want to sign up for your study.
Right.
Those are an important part of the dynamics of human life. Does your vision of systems biology incorporate those type of variables? So
It does. That's really a great question. And it does in several ways. One of the types of data we gather are personal traits. And we have lots of psychological mental tests that make assessments of the state of mind of the individual and how it changes over time. The second thing that I think is really going to be exciting is in the past year or so, we've developed for the first time ever a quantitative blood test that allows you to identify patients that have post-traumatic stress disorder. Soldiers from Afghanistan that are normal and have PTSD. And the reason that's important is it's the first time ever that a disease that heretofore had been defined purely by psychological terms could be defined quantitatively. My own absolute conviction is we will be able to get from the blood quantitative fingerprints that help us define wellness, that help us define different types of stress, that help us define a whole variety of different aspects of what's going on. And those fingerprints will just be reflections of the things that are going on in the brain. Okay, how about some other questions here?
Well, this could be addressed to anyone, but can one of you talk about how the results of all of this are communicated, and how do you see your targets of this research? And I assume you would essentially produce publications, and are the target of those publications physicians, or how does this get integrated into what?
I'll take a first stab. There's two answers to that. There's one that's a very practical approach that probably most of you would be interested in. So traditionally, when an individual is diagnosed with cancer, there's usually what's called a tumor board, where you get a number of experts together in the room and discuss that case from the different dimensions to try to arrive at a conclusion about the best course of treatment. And traditionally, that's usually been a surgeon and a radiation oncologist and maybe a medical oncologist and a pathologist and a radiologist that interprets, the CT scans and things. So what this systems biology, big data, and sequencing cancer genomes has allowed is another two more people at the table in what's now called a precision tumor board. And that is after sequencing an individual's cancer, you identify a number of the mutations or molecular changes that occurred in that tumor. And now you need someone with a biological understanding to now help interpret what drug, what target is now revealed. So with this precision tumor board, then that is communicated back to the patient in real time. The other person sitting at the table, which is quite interesting, that we've realized now is also very important. When you sequence the individual's, their tumor's genome, you also sequence and evaluate their constitutional genome, what they were born with, the quote normal. And we're starting, not uncommonly, to find things that over time have been linked to other diseases, not simply susceptibility maybe to that cancer, but to a neurological condition or to a cardiac condition. So now we need genetic counselors at the table as well to both counsel the individual patient and their family. So that's, I think, maybe a practical way of how this is starting to emerge, and we've been doing this now for the last 6 months around this prostate cancer project. And that overlays, I think your other point was that all of this information is assembled in publications that can be synthesized and evaluated by other scientists, but the data also goes into very large data banks that are anonymized, so individual patients can't be identified, but used for research to start collecting to see what's similar, what's different across different cancers, and then start matching these alterations with now new drug discovery, trying to hit those targets where maybe we don't have an existing drug. So, couple of ways.
Eric?
Yeah, I was going to add a minor addition, and that is it's sort of an ethical issue, and it's an expansion of something that Dr. Nelson just mentioned. That is that if you sequence the whole genome of somebody's normal DNA and you identify some of these traits that, who knows what it would suggest about you, and the patient didn't exactly consent to have those discoveries made. They didn't. And so, there's a lot of discussion about what do you do with that? I mean, do you really tell them that they didn't, And so this is an ongoing discussion amongst the ethicists and amongst the, what it really needs is it needs, society to decide what to do with this, because it's going to be more data than anyone ever really asked for. And many of these things are not going to be like, well, you're going to get this in such and such a date. What it really says is you're a little more likely to get this than someone else would be. And so do you really want to know that? I mean, so if you knew you were going to get diabetes next year because of this marker, then okay, you probably should know that. But what if you're only, twofold more likely at some point in your life to, something that happened to you that might be embarrassing? Do you really want to know that? I mean, I don't know. So, this is what town halls are for.
Do any of you guys want to comment on the FDA shutting down 23andMe? That seems like that.
I might just come up. I'm happy to talk about that, but let me just first say about the publication question, if I can address that first. So I think the idea of scientific publishing is, it's kind of this very traditional process where you do, in cancer commonly, these large clinical trials involving hundreds or thousands of patients. You put them together in this very formalized way. It's reviewed by external reviewers that are unknown to you. They usually tear it apart in ways, and then they make the presentation of the results better, then you publish it. And frequently, it's a year between the time that the manuscript's submitted and the time that it's published. And I think that whole model is really undergoing a dramatic transformation. First of all, there's an enormous lag. Secondly, the increment of learning comes in quanta of thousands of patients. And third, it's haphazard. Who reviewed your paper? What did they see? What did they not like? What did they miss? And it's subject to other things that you would rather not enter into the process. Although overall, I think it's a noble undertaking, and people do their best. I think what that is rapidly moving toward is a time where the barrier for publication is going to be reduced. People are going to publish things with much less of a time lag between discovery and the first time that it's disclosed. I think meritorious work will be work that is supported by additional work that people do, and the importance of findings won't be determined by 3 reviewers who happen to review your paper. It'll be determined by posterity, if you will, by how many other researchers built upon your findings and really fundamentally kind of changed the field. And I think that's happening now. With respect to 23andMe, I'm glad I got my 23andMe thing done. 99 bucks, I thought, man, that's hard to beat. And my wife's an oncologist, she had it done. She found out that she has a mutation in a gene called factor V Lysin that gives her about a 9-fold increased risk of getting a blood clot. So we go on the airplane and I say, Sibel, walk up and down the hall a few times. I found out that I'm more sensitive to Coumadin than a normal person. So, if I ever end up on Coumadin or this blood thinning medicine, I'm going to be more judicious about that. I found out my wife's more, if you give her a drug called Plavix, it's probably not going to work. And then I'm getting so that's all the medical stuff. I'm also getting all of these relatives that I find. And it's amazing. It's amazing. 99 bucks. And in the clinical, in the hospital, you want one of those tests, you're not going to get it close for 99 bucks. So I think the way this was approached was obviously flawed. I think the way the company approached their interaction with the FDA and their responsiveness to the FDA was obviously flawed. And they would have to acknowledge that. But I think the whole idea of trying to place constraints on the flow of information, that will not be a winning strategy.
I'll just add a word or two to that. I think so 23andMe is a company that basically can take a fingerprint of your genome with looking at 1 million features, and they could, as you heard, begin to make predictions about some of your predispositions. The FDA came down on them harder probably than the FDA has ever come down on anyone, really for 2 reasons. One is, like many people in Silicon Valley, they are infinitely arrogant, and they really look down the FDA, and the FDA doesn't look like that at all. And the FDA asked them for 2 things. One, to validate the nature of the tests that they provided, and that's really a complicated thing, but they could have worked with them and gotten something done. But second and more serious, they asked them to cease and desist in giving patients these statistical probabilities about the likelihood of disease, and I think for the most part, many of them are nonsense. They are really misleading in a lot of ways, and but again, they did not cease and desist. I think this it's going to be interesting to see whether 23andMe ever recovers, and I think they're really going to have a hard time coming back in the future. But I will say they did a wonderful public service to really bringing DNA to the fore in many people's minds, and it's what they've started is we'll be following up with this 100,000 wellness person study in spades. And of course, the question is, how can you identify things that are actionable? Now, let me say something about things that aren't actionable that you might have. I think there's an utterly compelling reason to want to know if you have bad news, and that is, for a lot of the bad news things, in the next 10 years, we're going to figure out how to deal with them. And if you know you have it, you can be at front of the line for the new approaches and everything. And this is a compelling argument, I can tell you, because in last August, 50 hoods got together and for about 2.5 hours debated whether we would have a family genome done. So the first big family whose genome is done. And we had all of the classical antis in the audience, but in the end, everyone agreed to have their genome done. So we're just finishing all these up now. And it's going to be very interesting to see. In my group, we had a long consent form. So in my family, I would say 80% of the people wanted to know everything. 20% only wanted to know the good news. So those are options you have. Okay, other questions?
So I actually had a different question, but the whole FDA thing brought up another thought. I just spent 3 days sitting in a conference with the FDA talking about this, specifically the IMDRF Medical Device Review Board, and the guidance that they're coming up with is kind of interesting and kind of addresses some of the things that you're doing. So one of the things that they're looking at is they're starting to find software that is designed to make medical decisions that is disconnected from physical hardware device as actually a medical device. So you're now essentially saying you're creating a system that's going to be giving you medical decision-making capabilities that, under these rules, becomes a medical device in and of itself. Furthermore, if you're personalizing the medicine, my question then becomes, aren't you now trading into the realm of each personalized decision-making process becomes an individual medical device that potentially has to be reviewed? And why this is important? There's actually some very interesting research coming out of Microsoft here, Microsoft Research, on a concept known as differential privacy. And essentially what differential privacy says is, if you have 10 degrees of freedom, 10 things that you're measuring, and you ask 10 different queries against that database that contains these 10 different measures, I can identify the patient. It doesn't matter how you obscure them, I can identify the patient. And the only way to address that is by actually introducing intentional error into the data set. So you now have this balance of how are you going to protect, if you're doing this individualized medicine, how are you going to protect the privacy in a way that conforms with what most people expect the FDA to protect you of, while keeping the data accurate? And so this goes to each one of you. You're each bringing a different approach to this, and so the question becomes, what's your approach here?
So my approach is that it is certainly true you can identify de-identified data from individuals. If you have an enormous amount of money, really fancy computers, and you know a lot of information beyond just the genome. I'm telling you about the genome now.
That's the Microsoft research on differential privacy says it doesn't take that much.
I'll tell you, have they done it on the genome? Have they done it on the genome?
N+1 queries.
I'd be skeptical about that. But I'll grant you that you can deconvolute anybody, but we're not going to do that most of the time. And I don't think most people really care. I think you'll go to the first level of de-identification, and I think that'll be absolutely satisfactory for most people unless they're infinitely compulsive about things. We talked about my family, and I told them, look, in principle, you can be identified even though you're de-identified, particularly because it's such a small group. I think most people don't really worry about that. I think it's the aficionados that say, gee, we can really do all these things. Let's have infinitely more rules and spend infinitely more money on regulation so we can absolutely be sure that we've convoluted, deconvoluted, put in noise, and done a whole variety of different things. And, you know, I think that's going to end up being an enormous waste of money. Maybe those people win. I certainly hope they don't do it for the taxpayers' point of view. But my own sense is that as we really drive to find solutions for a lot of these major problems, these things are going to become irrelevant over time. So
you may be protecting the data, but how do you know that 23andMe is? How do you know some random fly-by-night organization that's also saying that they're doing genetic testing is also protecting the data?
You know, I think you can only be responsible for yourself. And if the FDA wants to come up with rules and regulations, I think in the end it's just not going to happen.
That
It's
means
going
you
to
have to
be
be running the genetic test yourself, which most of us aren't capable of doing.
Oh, no. This 100K project, I think, can be managed very, very nicely in this regard. So,
I'm taking the prerogative of the moderator to move on to the next question.
yeah.
Okay, well, I have two general questions. One has to do with accuracy and consensus. You all have been talking about two basic objects, the human being and the disease object. Do you have accurate characteristics that can be used from the data collection side that you all agree on? And then the second part of the question is that once we get these relationships established, how frequently are we going to have to be evaluated to use the predictions that are available?
Well, I can just tell you with respect to this clinical trial that we have in breast cancer, you get all of this data and you're just trying to make sense. You get these computational people, you're trying to make sense of it. And DNA sequencing has some low error rate, and so if you find a change in a gene that you think might be important, it's an important question to consider. Is that real or is it just an artifact of a sequencing error? And one way that we address that is by getting multiple different samples of the tumor. If we find a change that occurs reproducibly across different bits of the tumor, then we gain a lot more confidence in the finding. We think that it's more likely to be real. One of the things that we try to gauge from our patients is, do you only want to know the stuff that we feel highly confident of? So if you have cancer and you're looking for a treatment, do you only want to know the stuff that where there's pretty strong reason to think that we're right? Or if we don't have any of those, do you also want information about the things that we're much less sure about? And And so far, it's my impression, we give patients questionnaires, we try to probe their understanding of these things. They seem to want everything that we have. They want access to everything, at least so far. And, but I think your point is really kind of raising a lot of, there is an enormous amount of ambiguity and an enormous amount of noise, and discerning true signals, definitely a non-trivial challenge.
You know, I might add that 2 weeks ago I spent half a day at NIST, which is the National Institute of Standards and Technology, and they're actually flying out in about 3 weeks and bringing a team, and we're going to be talking about standardization of how you procure your samples. That's really the first place to start. And then standardization of all the major data types, because these kind of projects that I described tonight will quickly spread all over the world. And what we'd like to do is set really rigorous standards for accuracy within the limits of the technologies we have today. And that's really, really important if we're to compare the data we gather in France and in China and in the US. That is, to know that it comes from similar kinds of samples with similar criteria for quality on the individual data types and everything. So, next question.
Hi. Actually, this is also addressing to Dr. Hood and Dr. Blau. What do you think? There are all these predictions made on treatment, and are there possibility of additional steps for validation of these all hypotheses with patient-derived assays? And
we've kind of done an interesting story. So we've enrolled 4 patients in our protocol. The second patient has, so we have data from our first 2 patients. Both of them have mutations that have never before been seen in breast cancer. First patient, very likely to be a driving mutation, and it's targetable with a drug that you wouldn't use in breast cancer normally. Second patient's more interesting. Second patient has a mutation in a gene that's been mutated, has been identified as a mutation in lung cancer. And in lung cancer patients that have this mutation, they respond to this FDA-approved drug. This FDA-approved drug is never given to breast cancer patients. But this specific mutation is different from the mutations that have been seen in lung cancer. It's like a single amino acid change, a tiny change. So when we found the change in this gene called ROS1, we sent emails to all of the ROS1 experts in the world, a guy in Taiwan, a guy in Germany, a guy at Tufts University in Boston. And it's like you send an email to people who are, and you get this like tutorial by the world's experts about everything they know about ROS1. And everyone answers, and everyone gives you this very kind of detailed response. So it's lovely. But the most interesting email was an email that I sent to a scientist at the University of Washington who sent it to his colleague, unbeknownst to me, who sent it to his colleague in Lawrence, Kansas. And this guy's described as, the way he's introduced to me is there are a few people in the world that know more about this than this guy in Lawrence, Kansas. And he thinks he knows exactly what's going on. He thinks that it's preventing this protein from being degraded. He thinks it may well be an activating mutation. Now, I can tell you what we're trying to do is in the lab see whether that's true or not. So it is possible to do these things, but on an individual patient basis, it's hard. I think what we're trying to do is convert these individual, and I think the challenge for all of us is to convert these individual patient experiences into something that's bigger, more important than an anecdote. How do you convert an individual patient's experience into real scientific knowledge? I think that's an enormous challenge.
And some of these, I've seen, at least in trade shows, some of these services being offered to grow that tumor ex vivo, just outside of the body, and then try to test different combinations on them. Do you see any possibility?
Yeah, we try to do that as well. So in breast cancer, it's hard to grow the cell. About 30% of the time you can grow cells. And one of our patients have been able to grow cells. We're doing it in leukemia, acute myelogenous leukemia, where you don't have to grow the cells. You just get them out of the patient. You put them in a high throughput drug screen to test susceptibility against a panel of 160 drugs, and then you get the patient on the drug that works the best. So Pam Becker is doing that work now, and she's done it in 10 patients, and it looks really encouraging.
I think I just want to add to that, that in some solid tumors, that's actually, it's not necessarily as predictive, because the tumor is actually living in an environment surrounded by what we call stroma, like blood vessel cells and inflammatory cells and many other fibroblasts and whatever. And the behavior of those tumor cells is very dependent on this niche that it lives in. And when you take those tumor cells out and put them on a dish, it doesn't always, in fact, it usually doesn't predict very well. So just so you know.
You know, I'll add just one more thing to it. A big concept that's coming up is the idea of doing a clinical trial where N equals 1. And the really interesting area that I think this is going to be done in widely to begin with is nutrition. Now, most of you may or may not know, but nutrition is in the dark ages. A lot of what we're told is generated from animal studies where there's little orthology between the animals and between humans. And in fact, in this 100,000 well patients that we're going to be looking at, we think by far the biggest set of actionable possibilities will be nutritional possibilities, and we'll be able, with each individual patient, to create optimized therapies and follow across time the reflections of success or failure of those therapies in the blood. Okay, next question.
I'm going to turn to a different kind of question. Some of you I know fairly well on this panel, others I don't. What was and each of you have devoted your lives to this work in a time in our country when people don't often hold the same area of focus throughout their work lives. So what was the spark, or who is the mentor behind bringing each of you into this life's work? And how do you stay motivated in an environment where each of you have referenced the fact that there's so many setbacks, there's so few times when you can really count on a discovery moving forward at a time when you really hope it will. So I'd begin with
So a lot of questions there. I can't say there was a particular person who did this. My personal career is in two pieces. As a clinician, I'm a neurosurgeon, and I operate on brain tumor patients. And that's it's fun to do. The short term, what you get out of it in the short term is really great. But ultimately, it's a very bad situation because most of those patients don't do well. And it's incredibly frustrating. And so the science is actually therapy for the ultimate outcome of the clinical work. And you know, there's no short-term gains in the science. They're long-term. They take, you know, we just got a paper accepted that my postdoc has worked on for 7 years. And it's, you know, it's a great paper, believe me, but it's, you know, it's a long-term success. And so if you balance these things out, short and long-term gains, you can, you know, I think the thing is that we all probably, I'm sure each of us will tell you that this is something which really matters. And you can do easy stuff, or you know, that really matters and it's easy, that's great, but a lot of stuff that really matters is not so easy, and you just do what you think is the right thing.
Yeah, for me, I don't think it was also a particular person. It was just more, during my training in medicine, we often would give drugs to patients for a variety of different diseases, not just cancer, and see this tremendous variation in response that really wasn't predictable at all. And so it was really I think what motivated me to try to study disease at a very fundamental level was trying to understand, again, this variation in unpredictability. And it was really the areas of oncology and hematology that I found were focused early on in understanding a true molecular mechanism underlying disease, whether it's something like sickle cell anemia, all the way through to the various cancers we treat. It's fundamentally a genetic disease, which has environmental influences, but at least it gave you a very strong foundation on which to build that was very logical and consistent. And it's not hard to get motivated, particularly when you see patients. There's just no question, because you see it in front of you every day. And the laboratory is fantastic. The challenges really are funding climate and administrative regulations. And those are the things that none of us were really trained for. We're not business people, really, other than Lee, who does it all. But you know, it's really, that takes us away from what we're really good at and what we really want to do.
I knew that I wanted to be a doctor like when I was in first grade. And then I read this book when I was like in maybe 5th grade, called Milestones of Medicine by Ruth Fox. It's out of print, but it talked about things like Walter Reed and Madame Curie, and Walter Reed going in and, you know, testing whether yellow fever, what the vector was for yellow fever by exposing him and his team to a bunch of mosquitoes. And it was so cool. And then I went to medical school. I loved that, and then did my residency. And I always knew I kind of wanted to do research, or wanted to give it a hard go. But I hadn't done research before I came to Seattle, and I was incredibly fortunate to land in the lab of a couple, George and Thalia Stamatianopoulos, who took a guy who was a pretty good doctor then, but knew nothing about research. And I spent 5 years there learning about in a field of research that has really nothing to do with what I do now, but learning how to do science. And had I landed in it, and so they were incredibly patient with me. It takes a long time. It's a very different world going from clinical medicine to going to a research lab. And if I landed in a different place, this very, very possibly wouldn't have worked out. And so I'm just incredibly fortunate.
So I had a high school chemistry teacher in Shelby, Montana, that to my enormous good fortune had gone to Caltech during World War II as a Navy meteorologist, and he decided any good student he ever got, he would send to Caltech. So beginning in my junior year, he started arguing. That was the absolute perfect place for me. I had more in mind liberal arts schools and things like that, but I did go to Caltech, and it transformed my life. I would have done something interesting, but Caltech was marvelous. The second person who really transformed my vision of science was my PhD mentor, Bill Dreyer. And he was an enormously creative, technically very adept kind of person. And he had 2 theses that I've lived by. One, he said, always do leading edge biology because it's more fun at the leading edge. And he said, 2, if you really want to change the world, invent a new technology. And that really would be transformational. The third example I would cite is my 2nd or 3rd year at Caltech, 72, 73. I read a book called The Structure of Scientific Revolution by Thomas Kuhn. And it was about paradigm changes. And they were in physics in this particular case, but what really came through loud and clear was, 1, paradigm changes really transform science, and 2, they really were hard to achieve, and you had to have enormous courage in the face of enormous adversity and people saying, well, that's nonsense, why would you ever try to do that? And to give you an example, when we were developing the automated DNA sequencer, I remember getting in an argument with Jim Watson, who said it was a crime that I was using public monies to invent an instrument when sequencing could be done perfectly well by grad students, and they could do all the sequencing they ever needed. And I pointed out to him that all my first grants to funding agencies, NIH had been turned down, so I was doing it with private money, not with public money. But he acknowledged later he was wrong, but it was a long time later. Okay, next question.
Yeah, it's very clear that a lot of the opportunities for advances will come with a lot more data. I mean, that's the motivation for getting 100,000 patients. But we probably should be thinking more like a scale of tens of millions and an ongoing basis and much broader. So I don't know what your thoughts are about how we scale this up massively, use of smartphones or other sensing devices. There are a lot of people, like one over here, who are more than willing to opt in, make their data available, so that then we could really operate at a scale on a continuous basis where much greater inferences could be made.
Couldn't agree with you more. I think that's absolutely a superb point. And what I see coming in medicine is a digitalization of medicine that's totally equivalent to digitalization of communications and information technology. And that means all these data are going to become dirt cheap. That means you'll be able to do almost everything on a cell phone or something that's equivalent. So I think the 100K project that I've talked about has one fundamental purpose, and that's discovery. What are the measurements that are going to be critical to optimizing health? Where I see really scaling this operation is creating a company that is going to use these insights in the most efficient manner and be able to scale it. And I'll argue in not too short a time, we can think about disseminating it to the developing world and have a democratization of healthcare that was absolutely inconceivable even 5 years ago. So I agree with you completely. And how we digitize medicine, and we need all the help we can get from all of you, is really going to be the critical question.
So I want to go back to my original question, which was, is my understanding that you've talked a lot about mutations in genes driving cancer, and that being a genetic disease. An understanding from, I don't know quite where I got it, was that by the time most doctors see patients, the cancer is sufficiently identified as a cancer. The cancer is sufficiently progressed that there are multiple mutations within the cancer itself. And so my question is kind of twofold. One, is this true, and in a way that obscures the base mutation? And secondly, is this part of what you're trying to do with the divergence to detect where that base mutation is occurring? And is it true across all the different kinds of cancers, or is there some similarity across the cancer?
So I'll make a simple, fundamental answer. In cancer, your normal DNA mutation rate increases 200 to 400-fold. So all cancers have enormous numbers of mutations. One of the incredible challenges is to figure out which of those mutations, or which combination of those mutations, are really driving the cancerous process, and are the targets against which drugs should be directed. Paul, did you want to add clinical experience?
I'll add a couple of things since we're right at the end. Different cancers are different, and some of them, if you look across the genome, are very messed up. They have all sorts of problems as the evolution of the tumor. And you could imagine there are many, many drivers all driving simultaneously. Late comers to the party are probably not very good targets for therapy. And sometimes it's hard to figure out even what the initial drivers are. And there's a lot of heterogeneity. And so that kind of a cancer may be maybe not the best choice for a single drug hitting a particular target. There's, on the other end of the spectrum, there's a handful of cancers that are really generated by an initial event that is really the driver, and the genome is relatively silent otherwise. And those, and CML is one of those, which is this drug that responded, this tumor that responded to Gleevec early on. But there are other tumors like that, and so you can imagine it's not like all cancers exactly the same. There may be a subset that actually would be very good candidates for a smart drug that would hit the original target if it was druggable. And then there's others that are going to be, have to another approach because it's really a bit like Dresden after the war. So it's just a mess.
Well, in closing, I'd like to thank the speakers, and I'd like to thank the audience for terrific questions.
Yeah.